Thank you very much for showing up. My name is Holger Becker. I'm from a company called Microfluidic ChipShop, and I'm going to talk about microfluidics today. And I thought this makes a much nicer slide to start off with than my boring official title. That's actually my boring official title. Microfluidics is a powerful technology for diagnostics and medical product development. And I hope you're somewhat familiar with microfluidics. Other than that, I will give you a brief introduction of what all this is about. Particularly as microfluidics as a concept has been around now for really quite some time. And of course, everyone is asking, what's now the commercial output of it? What can I actually do for my product? Or can I make use of microfluidics for my own product? And I think there are actually nowadays, particularly if you're talking about diagnostics, particularly biomolecular diagnostics, I think there's actually no new product development which does not include microfluidics these days. So the nice thing is we see that the commercialization is really progressing, mainly because nowadays you have industrial manufacturing methods based on the use of thermoplastic polymers. So thermoplastic polymers, typically all the polymer stuff around you, the technical polymers like COC, PMMA, polycarbonate, polypropylene, and so on. One of the drivers behind that commercialization is actually the increasing demand for a functional integration on chip. It also shows you that nowadays microfluidics has progressed to a state where you can actually make very, very complex microfluidic chips possible. And you will not only get these devices to work, you will also find somebody to actually make these for you also in high volumes. So this one here is actually one example of a fairly complex device. It's a sample in, answer out cancer diagnostic chip. It has on-chip reagent storage. It has amplification detection on a chip. So it's fairly complex. Another mover or another reason why microfluidics is increasingly used is actually people have been joking a lot about these miniaturization technologies and saying, well, this is not really lab on a chip, it's more a chip in a lab. You have a small chip and you have a lot of big instrumentation around it. But nowadays, because you can make this functional integration, you can actually now make fairly small instruments, ideally portable ones, where you can measure at the point of interest, maybe in a clinical lab, maybe in the field if you do environmental analysis or stuff like that. So low cost or ideally, even no instrumentation, everything is contained onto the single chip or on the single cartridge. That's possible these days. And of course, finally, everyone claims, hey, this is reducing cost. I honestly don't believe that. Cost, particularly in healthcare, is like entropy. There is only one direction. We can sort of maybe manipulate the slope a little bit, but I don't think that reducing cost is a real thing. Let's do a reality check, because of course microfluidics 10, 15 years ago has been hailed as the next big revolution. We're going to revolutionize the life sciences and diagnostics and the way analysis will be made. Well, if we look into where we are at right now, one of the things which I found really important is 5 years ago my talks, my first 5 slides would always be, why does it make sense to miniaturize? What are the advantages? What are the scaling laws and stuff like that? That discussion is through. Now, really everyone accepts, even in the sort of fairly conservative markets like diagnostics, everyone accepts the advantages of miniaturization. The scaling laws are known, and it's no big deal anymore. We see also a certain number of companies which have functioning systems on the market, and that number is actually nicely increasing, so the market is really taking off. Manufacturing technologies and infrastructure are basically available. That's also different from 5 or 10 years ago. Even if you have a very complex, fancy device made in an exotic material, chances are you will find someone to make it for you. We had a guy who really wanted to have microchannels in laser-etched calcium fluoride because he did some exotic spectroscopy. Well, he got it. And finally, the scientific base is really now broad and still growing exponentially. There's hundreds of groups in universities and other academic institutions working on microfluidics, so the know-how base is really going up a lot. But on the other hand, if you look at the amount of money invested, particularly in these early pioneering companies and the commercial output, so far there's a fairly gross misbalance. So microfluidics has certainly not been a field which has been investors' heaven. If you look at, let's say, the first pioneering companies like Caliper, Clara, Nanogen, and Orchid, these guys raised roughly about $1 billion amongst themselves. Caliper is the sole survivor with a current market cap of $250, maybe $280 million. So that's certainly one of the most critical points to consider when we're talking about Microfluidics, that from the investor's point of view, it has not been heaven. And that's now for the engineers of you, Microfluidics development can be really a pain in your bottom part. But of course, there are a couple of reasons why that's the case. The first thing, and that's really by far the most important message, Microfluidics is a truly multidisciplinary field. It's not just engineering or physics or microsystem technologies or biology or chemistry or material science. It's all mixed together. And you really have to combine the resources, you have to combine the know-how in all these various disciplines to really make a successful Microfluidics development. Several hurdles are possible, and you have to overcome them during particular upscaling, so from development through prototyping phase to volume production. And one of the things I'm always urging people is, because Microfluidics has a lot to do with materials and surfaces, to try and use scalable technologies right from the beginning, right from day one. And in this case, because we're talking about polymer as a substrate material for disposables, we really recommend use injection molding with standardized formats right from the beginning. There are lots of prototyping methods around where people think, oh, injection molding might be a bit expensive. I will show you later on why that might not be the case. But if you use a different material and a different manufacturing methods for your prototyping, and you would then like to scale up and make more parts of it, and you have to switch materials and technology, basically the results you obtain with your prototypes are just for the bin. A Microfluidic device which has been designed to withstand, let's say, with passive valves which withstand a certain internal burst pressure in material A, made with method A, and with a certain given surface roughness, will not work exactly the same if you do it with a different material, with a different manufacturing method. So eventually that valve simply might leak. So that's very important to keep in mind. One tendency we see a lot in Microfluidics is people tend to over-engineer. Oh, can't we make a 5-layer device with 37 integrated valves and stuff like that? Really bad idea if you talk about disposables, where eventually, even in high-value applications, manufacturing costs should be, you know, a few dollars a piece. Maybe Microfluidics is not the only field where that danger exists, but I'm a firm believer of the KISS principle. So make it as simple as possible, particularly if you're talking about disposables. Finally, also a message particularly for mechanical engineers. Please keep in mind that biology has an inherent variability. For example, if you design a Microfluidic device which works with saliva as an input matrix. For example, we've been doing things like that for recreational drug testing, for sort of criminal investigation work. The problem is, between me and you, and between morning and evening, and between having just had a fatty lunch and something like that, the viscosity of your saliva has a dynamic range of something like 20. So for some people it flows easily, for others it does not. And you have to design your device, your Microfluidic device, with that biological variability in mind. And that's pretty hard for a couple of engineers to grasp, that you can't specify everything to 5 digits behind the comma, and it's not going to change. So keep in mind that if you're talking about life sciences, you very often have biological variability in the system. But of course, there is hope. I wouldn't be doing this stuff for almost 20 years now if I wouldn't think that there is, of course, good reasons for doing that, and there are also ways of doing it the right way. So a couple of lessons learned, some the more hard way, some others not so hard. The first thing is, and that our clients usually do not really like to hear, there is no generic recipe, not a simple tick box list for development of a successful Microfluidic product. But there are hints available, and I think based now on our really mounting anecdotal evidence, I think we have by now a fairly good feeling of which projects are, let's say, hopeful and which are not. As mentioned before, Microfluidics is a truly cross-disciplinary field, and these complex tasks, you have to really map that cross-disciplinarity also in your team. One of the important messages is look at your target markets and look what is there already in standards like sizes for devices, like spacing of ports, like existing fluidity interconnects. I think one of the reasons why many of the early pioneering companies failed is they all built standalone systems which did not interface well with existing lab standards or lab workflows. So keep that in mind and try to make your Microfluidic system as easily integratable in existing lab protocols and equipment as possible. What we see is, and that's probably also a sign that the field is maturing, the material range is becoming increasingly heterogeneous. So it's not just an injection molded part or just a polymer part. It's a polymer part with integrated membranes and septa and on-chip reagent storage and metal or electrodes in general, sensors, silicon parts, whatsoever. So material range is getting increasingly heterogeneous and the size range is getting increasingly heterogeneous. Most of the Microfluidic devices typically are microscopy slide size, so 3 by 1 inch. So macroscopic part. It has microscopic features on it, starting from millimeter sizes like fluidic reservoirs through micron, let's say a few 10 to a few 100 micron sized channels, to nanostructured channel floors. So there are up to 5 orders of magnitude dynamic range of scale just on a single device. Again, that shows you how far that field has progressed. Despite the fact that nowadays modeling and simulation technologies have improved a lot, we still, for normal product development, see that there are several design iterations still necessary. We can do a lot of modeling and simulation, but the proof is in the pudding, so you actually have to make the stuff. So keep that in mind. You have to go through several design iterations. One thing which is really important is you have to cover a complete technology chain, and that's I'm going to show you in my next slide. And finally, my mantra, please, 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 please. For a design to manufacture, involve manufacturing people as early as possible in your design process. We had people coming to us with fancy 3D CAD models where they obviously have spent months on designing. You look at them for 5 minutes and say, you want this thing made for $3 a piece. Feature density, aspect ratios, minimum feature sizes, critical dimensions, tolerances, all off. Forget it. So involve manufacturing people early in your design process. Important. Okay, that's what I meant with technology chain. In this case, for the manufacturing of polymer-based Microfluidic devices. It starts off with the design, and that sounds very trivial, but it's not, because already at that point, you get that input from all these different disciplines. Application know-how, for example, even if you want to manufacture the devices, you have to know what this thing is supposed to be doing. For example, if you're looking for low-abundant proteins, or let's say if your client is looking for low-abundant proteins, you have to make sure that your surfaces are smooth and are coded in a way that you do not get unspecific protein binding, or your channel from your input to your detection zone is as short as possible to minimize sample losses. So that's important. You have to understand the application. Microfluidics, of course, you have to know the physics of microfluidics. You have to be able to calculate back pressures and distribution ratios and stuff like that. Material science, again, much is surface chemistry, so you have to understand what your material is doing to whatever liquid you have in there, or people use, say, laser-induced fluorescence as a detection technology, so you have to understand your optical properties of your material and so on. And finally, as I said before, manufacturability has an important influence on the design already. As we're talking about polymer replication technologies, the next step then is to make a replication master, which is a mold insert, the geometrical inverse of your final structure. And we typically prefer to do that with ultra-precision mechanical machining, which nowadays goes down to about, in theory, to about 10 to 15 micron. In reality, if you take tool wear and stuff like that into account, more to, let's say, 30 to 50 micron. But it can make arbitrary heights, three-dimensional structures, so that's our preferred method. If you want to go down to even smaller structures, you have to do lithography and electroplating. We can make very, very small structures that way, but you're basically limited to one height level. And then there are a couple of other fairly, let's say, more exotic microfabrication methods. We have access to all of them, but the first two we have in-house, and these are our preferred means. In the next step, then, you replicate that master, and I would say for our commercial devices, 95% of what we're doing is injection molded. We do hot embossing as well, and we do also casting, but I always tell people, particularly from academia who have been working with silicone casting, PDMS is bad. It's really bad. You don't want to use that in a commercial product. At that point, you have a microfabricated polymer part, and to make a functioning microfluidic device out of it, you have to go through all these back-end processing steps. The physical ones like drilling holes, cutting, dicing, sealing, assembly if you have filters or membranes in there or sensors, metallization if you want to make electrodes, and the chemistry-related ones like the immobilization of biomolecules, the tuning of hydrophilicity, on-chip reagent storage and stuff like that, and finally quality control. Something which is really important to keep in mind is that most people are concentrating in their development on this field here, because in prototyping, most of the cost, 80% of the cost, is in these early stages. However, if you really do product development and you would like to scale up your production to higher volumes, that's where the cost is, in the back end. And again, that's why it's so important to involve the manufacturing people in the design process, because by a clever design, you can save one of these back-end processing steps, or you can make these back-end processing steps easier, you will save a lot of money in the long run. So that's actually where the money lies and the value. It's in the back-end processing. It's not so much in the molding. Molding is only 10 to 15% of the overall value. Okay, how can we help you? So our company is basically organized in 4 different business fields: off-the-shelf catalog products, complete systems, customer-specific manufacturing and design, and collaborative R&D. A little bit on off-the-shelf products, because as said before, Microfluidics has always been seen as to be fairly complex. And if you're new to the field, you actually don't really would like to carry out an engineering project, which might take, $50,000, take half a year time. And because you're new to the field, there is a significant technological risk of failure involved. So instead, you spend a few $100s on these off-the-shelf devices, play around a little bit with it, get your hands dirty, make first experience in microfluidics, see how your assay is scaling in a reduced volume and so on. And only if you've done that, go to your custom design. Second important step, again, we try to use existing lab standards for our catalog products, so things like Titer plate, slide, microscopy slide, or CD as external formats, existing interfaces like Lure, Mini Lure, or Ollies for silicone tubing. All this helps to integrate these off-the-shelf components into existing equipment. We do complete systems, so not just the chip, but a chip plus the instrument, plus accessories and everything else. So a few examples here: a continuous flow, PCR, instruments, so basically a platform for thermal control, coming with a variety of chips, capillary electrophoresis system with contactless conductivity detection, basically for food and water analysis. And as I said before, it's not only instrument and chip, but it's also the accessories coming with it, pumps and so on, plus these little support kits with bits and pieces you actually need to run the stuff like tubing and oil and things like that. And finally, we have an application group in-house, which also helps people develop protocols for that. So in our systems, all the systems come with starting protocols so people can really start working with them. Biggest part of our business is customer-specific development and manufacturing. And as said before, one of the things I really think is important, that you use scalable production technologies right from the beginning, and for us that basically means injection molding. To keep development cost and cycle time as small as possible, we use family tools, so we have a fairly broad range of sizes and interfaces available. With exchangeable mold inserts, so typically for your first couple of development steps you would only have to invest in a mold insert, which typically is about an order of magnitude cheaper than a complete injection molding tool. One of the things Microfluidics really suffered from was over-promising. So I always urge people in saying, be realistic in your expectations. Be realistic what you tell your boss or your investor. And as I write quotes typically for 1 million or 10 million parts every second week, I always tell people when they say, oh, do you have the capacity? I say it's easier for me to make 1 million parts than for you to sell 1 million parts. Saying or meaning that it's actually not the fabrication which at the moment is the bottleneck, it's the markets. A few examples here. That's a nanowell plate with organic semiconductor photodiodes. So that gives you an example of a very hybrid system with respect to materials. That's actually a very nice one for drug discovery, or it's a chip for dielectric cell assembly. So we do liver chip co-culture. You can actually sort of grow liver tissue in these chips, and they behave like in vivo. And a final one, that's a cartridge, a diagnostic cartridge for agglutination assays. Again, a fairly nice one, because in this case, that really makes original, let's say that takes advantage of the original advantages of miniaturization. In the normal cartridges, you have a gel, which is antibody coated, which you have to first to fill in. It limits shelf life. It has problems in sort of keeping everything upright, it's fairly expensive to fill in. In this case, this part has a multiply bifurcating network of microchannels, again, antibody coated. And if you have a positive reaction, which is an antigen reaction, agglutination, a blood clot forms, and it just blocks the microchannel. And it comes for free because it falls like that out of the injection molding machine. Very nice. Finally, we do a lot of collaborative R&D projects, either with industry or with the academic community, because microfluidics is still a scientifically hot topic, and it's still progressing quite rapidly, and we would like to be on the cutting edge. We typically generate between 3 and 5 publications a year, but more importantly, we're a fairly small company, about 45 people right now, and we can't do everything ourselves. So we have to create these sort of networks of excellence. We have to partner with people who can fulfill certain tasks, and I think we're fairly good in these collaborative efforts. And of course, for clients, if we can cover certain aspects of a development with these publicly funded projects, that helps to reduce development costs. A few examples, that's a fairly complex multi-sensor platform for cancer diagnostics. As I said, sample in, answer out device, which currently is in commercialization stage. And that's one of these examples which shows you having nanostructures on the same device as microstructures on a macroscopic device. So you can see that is sort of a very simple test structure. So this was more basic research. It has microchannels, 50 by 50 or 100 by 100 micron. And on the floor of these microchannels, you have these nanostructured sawtooth, which have a period of 1.8 micron and a height of 200 nanometers. The idea there is that most of the thermoplastic polymers are hydrophobic, but you would, for particular diagnostic cartridges, you would like to have self-filling devices. And by structuring that floor, you can actually get self-filling even in fairly hydrophobic devices. The nice thing is, this again comes for free in the molding process, so you would save a back-end processing step like plasma activation or plasma treatment of a hydrophobic material. So again, clever design saves you money in production. Okay, let me conclude. Something which is important, microfluidics really needs attention from both sides, not only just the technology, which actually has received a lot of attention, but also from the business side. So as I said, it's easier for us to make 1 million parts than for you to sell 1 million parts. Polymer microfabrication has its challenges to the large variety of materials and different methods, and it's much less standardized than the silicon world. But in the last few years, process know-how has really increased exponentially, and manufacturing capabilities have been set up. So there's actually, I don't think that you will not find anyone to make these devices for you. And I always tell people there's no reason not to use polymers as a material unless you need semiconductor functionality. That's the only thing where silicon still makes sense. Second conclusion, we try to bring microfluidic systems into the market. I'm not claiming we're the next Intel of microfluidics, but we would like to see them in the daily laboratory life. So we make components easily available through our catalog. We work with existing standards as much as possible, easy to handle components with on-chip fluidic interfaces. We have certain platforms in standard formats. We provide complete systems if necessary. Of course, we try to be better and cheaper than anyone else. Oops, my last slide for some reason seems to be missing. On my last slide, it should have shown you actually a Segway. A Segway was supposed to revolutionize urban transport. And of course, I don't know how many of you ride with a Segway to your work every day. I just want, sort of the message is revolutions take time. Microfluidics was supposed to be revolutionizing the life sciences 20 years ago. I think we're seeing the first steps towards that, but just keep in mind revolutions take time. With this, I would like to thank you. I think we have a few minutes for question and discussion. We have a booth in Hall B, 3294. If you would come up, we have several examples and material there if you'd like to discuss. And thank you for your attention. And again, thank you for preferring me over taco or burger for lunch. Thank you.